we're going to make an agarose gel and then run the gel. So this process is called agarose gel electrophoresis. First we need to assemble our casting tray. So the clear little piece there is the gel casting tray, and we put it into the other component, which is the other bit of the casting tray, which seals in the ends and stops the agarose leaking out. So we need to make a decision here with which comb we use. Comb is also known as well former. So here we're going to run 8 samples, and I have enough space for 8 samples, so we're going to use the 8 comb. Okay, so our casting tray is ready, we've got our comb in, and now it's time to pour the agarose. So the agarose has already been melted in the microwave and cooled down to a nice comfortable pouring temperature, about 60 degrees. The agarose is made up in TBE buffer. Very important not to make up the agarose in water. We need to use the correct buffer, otherwise the current won't run. Also, very important here, remember to add our DNA stain. In this case, we're using the gel green dye. So we need to remember to add this into the agarose so the DNA picks up the stain and we can see it under UV light at the end of the procedure. There are lots of other possible stains we can use here: gel red, thiazole orange, ethidium bromide. The main thing all these have in common is that they bind specifically to DNA and they fluoresce under UV light. Don't need to add very much stain. In this case it's 10 microliters, and that's of an already diluted stock of gel green. So these are very potent dyes. You only need to add a little bit, and that's into 50 ml of molten agarose. We're going to mix it just by gentle swirling. Don't shake it, because if you shake it you'll get lots of bubbles which will cause problems later on. And we're going to pour the agarose into the casting tray in one smooth motion. We don't want to do it in lots of little dribs and drabs, or else again we can get the problem of bubbles forming. Also then the agarose may run down the outside of our bottle and make a mess. And that's it. Now we just need to wait for it to set. So it generally takes about 5 to 10 minutes for the agarose to set, and then we can put it into our electrophoresis tank. So we're going to disassemble the casting tray apparatus. So loosen that screw knob, pull out our agarose gel in the casting tray insert, and now we'll get our tank ready. So we take the lid off the tank and we're going to get it set up in front of us. Usually we put the black terminal on the left, so the negative on the left and the positive on the right, and we'd run it from left to right. So if we're going to do that, it's very important the samples are up at the negative end and they're going to run towards the positive end. Easy way to remember this is to remember run to red. Okay, next we're going to add the buffer. Now very important that this is the same buffer that's actually in our agarose gel. So in this case it's TBE buffer. So we pour it into each side of the tank and make sure that it comes up just enough to cover the surface of the gel. But we don't want to put an excess amount of buffer in, just enough to cover the gel is the correct amount. Note that we haven't taken our well form or our comb out yet. The reason for that is that when you take the comb out, there is a risk that the wells can collapse on themselves. So if we do this in the presence of the tank buffer, it minimizes the chance of those wells collapsing. So we're being very careful here about pulling that comb out. It's worth taking some time and being careful with that so that you don't wreck the wells. These are fairly fragile. Okay, before starting we need to make a plan of what's going to go into what lane on the gel. So we've got 6 lanes here that are going to be loaded. The first one's actually left blank. Then we're going to have our DNA marker, and then 4 other samples which are the DNA samples. It's typical to leave the first lane blank. Sometimes the very edges of the gel run a little bit wonky, so we want to put the samples towards the middle of the gel. Okay, so this is our loading dye. Very important not to get this confused with the actual DNA stain. So one of them actually binds to the DNA. The other one just provides a colour that we can use to track the progress of the electrophoresis. The other thing that this coloured dye contains, the loading dye, is a density agent, something like glycerol or phycol or sucrose, and this helps the samples to fall down into the wells. So it has 2 purposes. One is to track the samples during electrophoresis, the other is to ensure they get down into the wells. And what we do is we set up a couple of little dots of that loading dye along a piece of parafilm, and then we come back and we mix our samples with that loading dye. So here we're loading some PCRs, and you can see these are little PCR strip tubes. So we're going to open up the tubes. At this stage, we don't have to be too concerned about sterile technique with our PCR tubes. The PCR has already run. All we're doing here is just loading the samples onto the gel. So we need to adjust our pipette to the correct volume. And in this case, we're going to load 5 microliters of PCR. That's pretty standard for a PCR. So if we just put those PCR samples straight into the well, they wouldn't go into the wells properly. So we need to first mix them with the loading dye in order to get that to work. First thing we're going to load though, before we get to our PCRs, is our ladder or marker. These both mean the same thing. It's a series of standard size DNA fragments that we can use to compare the size of our PCR product to. So we had a little bit of that 100 base pair ladder, and we're going to mix that up with the first dot of blue loading dye. So we just mix by pipetting up and down a few times, and then we suck up 5 microliters of that mixture, and we're going to load it into the first well. It doesn't matter if there's a little bit left behind, there should be plenty there getting loaded into the gel for us to see a sample at the end. So note how she's using one hand to steady the end of the pipette while the other hand squeezes the plunger, so that's good pipetting technique. You want to steady that business end of the pipette that has the tip on it to get better control over the loading into the well. And she's doing the same thing when she's mixing the samples with the loading dye, so just steadying the tip with one finger and then sucking up and down to do the mixing, and then over it goes into the wells on the gel. It's okay to touch the yellow tip here once it's come out of the box. So in this case, if the tip gets non-sterile after use here, it really doesn't matter. So this is not an aseptic technique like a streaking a plate. This is just a molecular biology technique, and it doesn't matter if a few bacterial cells get onto our pipette tip here before we load the gel with it. Note though that she is changing tips between every sample. That is important. We don't want to get contamination between our samples in case we want to come back and do something with them, like say clone or sequence those products. Here we've just got a zoomed in view of loading the gel, so you can see the sample with the loading dye, and we're just slowly squeezing it down into the well. Note how it falls into the well on its own because of that density agent. You don't need to put the tip right down the bottom. You can just have the tip hovering above the top of the well, and the sample will fall down in due to its high density. Okay, so all our samples are loaded. Now it's time to put the lid on. Very important to get the lid the right way around, so the black terminal must connect to the black electrode and the red must connect to the red. And same when we're plugging it into the power pack. Black has to go to black and red has to go to red, or we can get an unpleasant surprise later down the line when our gel runs backwards. Many of these modern power packs will actually not let you connect them up the wrong way, but it's important to be mindful about this because some of the more old-fashioned apparatus will actually let you connect it the wrong way, so you need to be careful. So everything's connected up. The next thing to set the voltage, we're going to run this gel at 180 volts. The choice of voltage depends on the size of the gel, the volume of buffer, the size of the product, a whole range of other things. 180 is pretty standard for a small gel like this. Okay, then we just hit run. Okay, the gel's now running. Often a good idea is to check for current, which is milliamps or MA, and we can see we've got some current there, which is good. So that means everything's working as expected. She's just going to have a look now and just check that we're getting bubbles forming on the electrodes, and that's another good just confirmation that the gel is running, that we have current running through the gel. We've remembered to switch it on and all those other things. So here's just a zoomed in view of the electrode, and you can see the bubbles coming off the electrode. Depending which electrode we're looking at here, it could be hydrogen or oxygen, basically just electrolyzing the water. Okay, so we come back after about 15 minutes to have a look, see if everything's run. It may take a bit longer, anywhere between kind of 15 to 45 minutes would be standard for a gel this size. Check the loading dye and make sure it's gone far enough down the gel. Switch off the apparatus before opening the lid, very important. We don't want to give ourselves an electric shock. And then we're ready to take our gel over to be examined under UV light in the transilluminator. So we just drain off the excess here. Note that she's holding the gel in place while the liquid drains off. And you can see here that the dyes have progressed down the gel. So in this loading buffer, the pink dye goes about 3/4 of the way down, and the blue dye goes about halfway down the gel. And that's about far enough to run for a fairly small PCR product.